This is a GCSE presentation looking at the food tests. To get started, you've got three questions. You've got to identify the three main food groups that animals need to survive. You need to name the enzymes that are involved in the digestion of these food groups. And then you need to come up with as many specific examples of substances in the food groups and the related enzyme that does the digestion of them, identifying their products. Pause the presentation now, and when you're ready to go through the answers, you can restart the presentation. So pause now. There are three main types of substances that you need to be aware of. You've got your carbohydrates, your proteins, and your lipids. The enzymes, you essentially change the ending of each of the different reactants by adding ASE on the end to get the names of the enzymes. And you need to know what they're broken down into, the products. The carbohydrates are broken down into simple sugars, quite often glucose. Proteins are broken down into amino acids, and lipids are broken down into fatty acids and glycerol. In terms of the specific digestive enzymes, there are quite a number of them, but these are the ones that you should be able to describe based on what you should know at GCSE and digestion. If you are not aware of this, I'd suggest you go and watch the digestion presentation again and go back over this and make sure you memorize it. Any of these can specifically be asked about in the GCSE exam, so make sure you are aware of everything on the table. In this lesson, we're going to go through the food tests, and we're going to identify for each food test what you need to do the test and what a positive result would look like. There are four substances that you need to be able to talk about, and there are 5 different tests that they could talk about in the exam. At this point, all you have to do is match up the name of the substance with which test you would use. Pause the presentation now, go through your answers, and then when you're ready, we'll check to see how you've gone. So pause now. In terms of answers, a sugar test, what we would do is the Benedicts test. To test for starch, we would do a starch test. To test for protein, what we do is a Biuret test. And lipids, we actually have 2 potential tests. The most common one in the GCSE exam is the emulsion test, but it is not impossible that they will ask you about the Pseudom3 test as well. So I would strongly recommend that you are aware of that one too. But if you're asked to describe a test, the emulsion one is absolutely fine. So what do we do in a Benedicts test? Well, a Benedicts test is a test for sugar. It requires Benedict's solution and a source of heat. Often we talk about a water bath in the form of either a beaker with water in it on top of a Bunsen burner, or of course one of the electric plug-in water baths, whichever way you want to do it, but you do need a heat source, otherwise you will have no reaction. And if you have sugar in the substance that you're testing, the Benedict's solution, which is blue, remember you have to say Benedict's solution, just Benedict's isn't enough, and it will change from a blue color to either green, yellow, orange, or red, depending on how much sugar is involved. I strongly recommend that you learn the color sequence, green, yellow, orange, and red, particularly if you are planning on looking into A-level biology. You should know those whenever you start your A-level biology. It'll give you a head start. And that's the Benedict's test for sugar. The starch test is where we take a substance called iodine. And iodine is an orange-brown color. And if it is tested and we have starch present, then it'll turn to a very dark blue or black color. For a protein test, we do what is called a Biuret's test. Now we can use a Biuret solution. You can have Biuret solution as one substance. You can also have Biuret solution in 2 parts. And quite often you'll have to shake this test for it to get it to work properly. If you use a Biuret solution that's an all-in-one single solution, what will happen is you will have a purple color. If, however, your Biuret solution comes in 2 parts, part A and part B, you will find 2 distinct layers, a top layer and a bottom layer, and in between that you will find this layer of purple, and that is how you have a positive test if you have a 2 parts Biuret solution. In the exam, they normally talk about all-in-one biuretes, but just in case they do present you with the two-part one, you're still looking for that purple color, but it'll be a band in the middle between two other layers. The last food group we're going to test for is lipids, and there's two tests. The first of these and the most common one in the exam is the emulsion test. And in the emulsion test, we have to use equal parts of ethanol and equal parts of water, normally 2 centimeters cubed, but as long as it's in equal parts, that's the important thing. And if there is lipid inside the substance we're testing, we will have what's called a milky white emulsion, and you have to use that wording to get the mark in the exam. The second test that involves lipids is called the Pseudom3 test. And a Pseudom3 test, we get equal parts of water and the test substance. And we just simply add in a few drops of Pseudom3. And if we have a lipid present, we will have a red layer on the surface of the water. One of the things they may ask you about the food tests is they may ask you about risks involved or hazards involved and how you can stay safe. So at this point, pause the presentation, come up with things that you can do to stay safe when doing any of the food tests. Some of them are specific to certain tests. Some of them will apply to any type of food test that you do. Pause the presentation now, and when you're ready, restart and we can look through your answers. So in terms of the food tests, here's the safety things you need to be aware of. For all food tests, we need to wear safety goggles. That should be your go-to answer. We also need to keep the chemicals away from our skin and eyes, and we wash accordingly should we come in contact with any of the substances. When we're talking more specific, Benedicts Test, the Biuretes Test, and the Starch Test, we can all refer to them as being an irritant. So those solutions and chemicals are actually labeled as irritants. Note if your biuretes is a two-part solution, one of those parts is corrosive and the second one is an irritant. In terms of the other tests, they're actually using substances that are flammable, and as a consequence we need to keep them away from flames and all the things that we'd normally do to protect ourselves from any fire from starting. So to finish off, you're going to unravel a bunch of anagrams. They're all related to the lesson. Just be aware all the answers will come up at once, so make sure you have a go at all of them. Spend a little time onto it. Do spend a bit of time onto it, but no more than 10 minutes. If after 10 minutes you can't work out what they are, it's not a problem. Check your answers and then you can go back through the presentation if you need to find out more information. So pause the presentation now. Hopefully you're able to get all, if not certainly most of the anagrams, and here are the answers. And they're all linked in with the presentation. Again, if any of them don't make sense, or if you're not aware of where any of them have come up during the presentation, make sure you go back and watch again so that you're aware of the connection.